Hi, welcome back to Colloquium. It's my pleasure to introduce this week's speaker is Huy Dinh. Let's see, Huy began a career in computer sciences at the Vietnam National University in Hanoi, and then did a master's there as well before doing a PhD in bioinformatics at the University of Austria. After that, we came to California, and at the University of Southern California did a postdoc in epigenomics. And I think the lab moved partway through that, right, to Cedars-Sinai Medical Center. And then we did a second postdoc at La Jolla for immunology. So altogether, I think what's interesting is how many different types of environments you've been in over the course of your career. And all the while kind of moving in the direction of some really exciting work that we're going to hear about today, which, I always love hearing about heterogeneity, because I think there's really just unappreciated levels of heterogeneity in biological systems. So what we're going to hear about today is a multi-omic single cell approach to understanding heterogeneity associated with immunology, immunological cells, and links to cancer. And I really look forward to hearing this. Thank you.
Okay, thank you. So I hope everyone hearing me on Zoom. Thanks for the opportunity for giving a talk here. And so just like thanks for the introduction. So I joined here in 2020. So this is my third year, and I have a joint appointment with oncology and biostats, and that because of nature of my training. So we're trying to do both. We're not good at both, but we're trying to do both. And I'm gonna show you the approach that we're doing today. So today I'm gonna talk about our analysis using single cell and spatial profiling data. What we're looking at are the cell-cell interactions, and we think that may help us to identify the marker in inflammation and cancers. So because part of it is I want to be a trainer in genetics, so I want to tell you about my training background. And as Nico introduced, I have a background in computer science, where I did BS and BAs in computer science. And my master thesis is to solve the phylogenetic problem that I think many of you are familiar with. And then I moved on to do a PhD in bioinformatics. At that time, next generation sequencing started booming. So I developed methods in whole genome bisulfite sequencing, which were used to profile DNA methylation, genome-wide DNA methylation that some of you may be familiar with. And chip and chip-seq. No one use chip anymore, but at the time that I use as chip, and the method also work for chip-seq as well. And then I moved to US to do a postdoc. I did a postdoc in LA, but across two institutions because the lab relocated. And that is where I start involved in cancer data, where we use multi-regions. At that time, we don't have single cell. In order to study the heterogeneity, we sequence different location of the tumor, right? At the core, and then we compare. We published a number of papers in esophageal and liver cancers. And then one of the project we published later is we discovered the sequence context of global DNA methylation loss using TCGA DNA methylation data and machine learning methods. So and then I did my second postdoc. So for the trainee in the audience, so doing second postdoc is not bad at all, because you can learn new things. The reason I did second postdoc is my lab moved to Israel, so I want to stay in US, and I did a second postdoc. So I learned a lot about immunology, and at that time, single cells start being something that's very powerful. And one of the things that helped me to appreciate the technology is it helped us to identify the previously unknown rare subset. This is an example. This is a paper that we published about before I came here, where we discovered the progenitor of neutrophil progenitor in human bone marrow. We use a technology called cytoff or mass cytometry. Basically, it's flow cytometry, but using a mass spec, and then you can profile up to 40 markers instead of the 6 or 7 markers like the normal flow cytometry. We are able to identify the progenitor, which make up 0.1% of the cell in bone marrow. And then we use single cell analysis. We profile about 58,000 cells for the fallopian tube, which is widely accepted as a tissue of origin for the common high-grade cell ovarian cancers. We are able to identify the early epithelial subset with less than 200 cells. And if we don't have 12 samples, probably we're not going to be able to believe this subset. So we are able to link them with the gene signature from TCGA, and they are linked with the good prognosis subtypes of ovarian cancers. And another example is we are able to identify biomarkers that make up about 5% or 7% in the tumor-associated fibroblasts and 1% of the total cells. And they also link with the prognosis marker here. So this is a paper that I published before I came here. And when I came here, I'm very interested in immunotherapy. So, and as you hope you know, that is considered as the breakthrough of the year by the science journals. I believe it is before the Nobel Prize was awarded. So briefly speaking, they discovered the way to use T cell to attack cancer cell. Right, so I'm intrigued by just the immune system, right? So like just like it's in our body, in blood, and it can use to kill cancer cells. That's very intriguing. However, we have a problem. So only 20% of patients respond, and that is depend on cancer type. One of the things with immunotherapy is if it responds very well. Some of the patients like considered cured, right? But if both of them like don't respond, so we want to improve that. And it depends on cancer type. Melanoma have a very good, next to 50%. Non-small lung cancers and maybe other cancer have around 30, and other have 20. But many cancer type doesn't have response at all. For example, prostate cancer, breast cancer, some type is also very hard. So we are giving the heterogeneity matter, the tumor immune microenvironment matter. And in the field that we just simply consider there's 2 types of tumor immune microenvironment, hot tumor and cold tumor here. And you can see like the hot tumor are linked with, normally linked with the responder, and the cold tumor is with the non-responder. And in our lab, we take what we call is big data resolutions, big data solutions. And the big data is really booming since the Human Genome Project, as we all know. But this is with where they sequence up to like 30,000 cancer patients is also play important role there. Like a lot of machine learning or other methods developed based on TCGA data as well. And recently we are have the single cell and the spatial data. So I think that's the best time to do data science in cancer immunology right now, and for the upcoming years. So I think it's really good time. So immunotherapy use T cell to attack cancers, but T cell is not a branch of immunology. So if you have the basic immunology class, you know there's other branch of the T cell, like myeloid cell or innate myeloid cell and other response, right? So T cells still like the main focus of the field. However, in 2020, it is one of the recap of the cancer research institutes where they summarize AACR, which is an annual meeting from the Cancer Association. Now we start appreciate as a non-T cells component, especially the so-called myeloid cells. So this is a work that published by Krumel Lab from UCSF. So when they did a lot of RNA and flow data, even not single cell yet, and they have a lot of sample here. You can see one of the things they found is when they look at the 3 features, they have immune rich, immune poor, or T cell centric. But if you increase the feature in, and you start seeing what they call is immune archetypes, meaning it's not just T cell, but also others compartment going to contribute. So things are much more complicated than just T cell, which driving the anti-tumorals in tumor microenvironment. So from our own experience, we also see it from immune cell called neutrophils. So this is the work that we published where we discover this subset of neutrophils which have the pro-tumoral, meaning it suppress T cells. So this is a subset, and you can see like they decrease the T cell activation marker, which is the CD69 here compared to other subset. However, we discover another subset as well. It could be new here, and they are able to stimulating T cell. So within a myeloid cell type, we call neutrophil, there's both pro and anti-tumoral, and they're working with T cells to modulate how the tumor microenvironment going to respond to the treatment. So today I'm going to talk about cell-cell interactions, and I don't have to convince this audience. They are the fundamental for developmental and pathogenesis. So this is the community where we have they call it multicellular community, right? And the cells are not stand there alone. So they interact with each other, and this is one of the most famous cell-cell interactions since the born of the immunotherapy, which is a PD-1 that PD-L1 that is expressed in tumor, and PD-1 is in T cell. Right, basically PD-L1 bind to this and prevent the T cell from killing tumor cell. So T cell should be killing tumor cell because the cells off. However, so because this binding is not. So, but if we block this binding, so T cell are able to attack the tumor cell again, and that is the basic of immune checkpoint therapy. So there's a different way of study cell-cell interaction, like using imaging or labeling the cells or using the functional. And today I'm going to talk about the single cell approach. So this is the outline of my talk. So I'm going to talk about an integrated approach where we use single cell and spatial profiling as well as bioinformatics to identify cell-cell interactions. And I'm going to show you 3 examples, it all unpublished. So we saw the interaction in myeloid cell with marked humans burn severity or progression. So the work in collaboration with Anna Hatenocker lab here. And the second work is we saw we identify marker with immune checkpoint blockade response in head and neck cancers. The work was in collaboration with Paul Lambert lab and the head and neck spore community here. And the third is we identify markers that potentially can predict the early high-grade serous ovarian cancers. And that paper was in review right now. So now I'm going to talk about our approach. So in our lab, we build a framework that we call it is bioinformatic analysis of cell-cell interactions that could inform the biomarker or the therapeutic target. So we all start with the discovery data sets, mainly single cell data, but also we use a high dimensional cytometry for a certain type of immune cell that hard to get single cell. We generate the data ourselves, but we also took a lot of publicly available data out there. Since single cell doesn't have spatial information, we also use spatials. Also, we recently have the data where they have both, but it's still very limited. And the platform we usually use 10x, we did with the biotech center here in Chip Lab and also as a platform as well. And my previous work mainly in humans, but since I have my independent programs, I more appreciate about the model organisms, because I want to figure out the mechanism and what happened with what I found in human data. So we utilize zebrafish in collaboration with Anna, and we using the mouse model in collaboration with Paul Lambert from Oncologists. So this is the analogy that I like a lot so you can see like this is how book RNA-seq look, right? It's like the logo and then the Lego, and then you can see like all of them mixed together. That is what you have with the TCGA data, right? So you can see like it is very under appreciate the heterogeneity within the data. Single cell is here, right? That gives you like different cell type and how abundant they are within the expression. Spatial data give you the expression. You can see like this. However, spatial data, the best resolution is the spot that have like about 10 or 20 cells. Right? This is not single cell yet. What we need is this. Right? We need the one that have both. So like that is and I think we're gonna go towards that very quickly given the development in the technology field. So in our lab, we focus on what we call cell-cell interaction here. So we want to infer the two cell type in which ligand expressed in one cell type and their corresponding receptor expressed in another cell type, right? And this is pioneered by Sarah Teichmann lab from Sanger Institute, where they have the cell phone DB2 and a number of other tools come out from a different lab as well. And so you can see like basically they ask if the mean of the expression in cell type A and the product between the ligand and the receptor. And if they are not observed by randomly, such as like if they permute the label of the cluster in the single cell, right? And this is only account for one pair of ligand receptor. So recently there's a work come out from UCSD where they account for the interaction between two types of cell, but account for all ligand receptor, right? So this is an example where they use a binary score where they threshold and say what if the ligand expressed or if the receptor expressed in the certain cell type. And then they have the score like this, and they are able to infer the subset of ligand receptor pair. We have the interaction score best correlate with, for instance, like cell-cell distance by imaging. So after have the cell-cell interaction, we use imaging in order to validate this and also going further. So we use a vector imaging system from Chip Lab here, where we can validate a certain marker there. And we also take advantage of the technology called Visium from 10X. We work with the Chip Lab and Biotech Center here to do that. We recently explored the technology called COSMX, where they did both. We have spatial and single cell resolution for the same cell. However, COSMX still limited on 1,000 genes. You don't have the whole transcriptome yet. But I think the technology is gonna scale up very, very quickly. And because of the zebra fit work, we also want to incorporate the microscopy imaging, especially for light imaging, which we can do with fish in order to integrate with genomic data. So in our lab, after we identify this, my bioinformatics student usually go this way. They took another data and they asked if the ligand receptors interactions are specific in one condition, for instance, healthy versus localization, normal, or tumor here. And they also incorporate both of the data, asking if the ligand receptor is spread in the same position in the tissues. And since the data is still limited, we also computationally decompose them using book analysis from publicly available, or we generate them ourselves so that we can scale up into 100 samples or something like that. And then try to link the score we found here with a clinical feature where we have a higher number of samples. And my experimental students usually go this way. So they want to validate the marker using ISC or IF, the immunofluorescence assays, or they do the cytokine profiling, because that is one of the typical signaling that we got from here. And some of them took the two cell types that they're interested in with a specific marker they identify from single cell, and then co-culture them and ask what happened in vitro. And I have a student who are good at mouse model, and she used the mouse model to try to inhibit the signaling by ligand blocking antibody or just CRISPR out the receptor and ask what happens. And down the road, we want to incorporate the gene regulatory and signaling pathway in, because what we saw here are just upstream signaling. and we want to add epigenetic, metabolism, and morphological and behavior that we can identify from imaging image. So we want to ask whether the myeloid cell type, their plastic and their interaction can predict disease progression and respond to certain treatment. So now I'm gonna use the first example where we use this approach for the human burn severities project. So we work with a burn surgeon here, and this is another paper that where we see most of the burns are categorized as severe, not severe, based on one of the common way to use is the TBSA, which is total body surface areas, and you can see like here. Here's the statistic in North America, and most of the burn are actually less than 20%, and some of the burn are more than 20%. Right, it's very few. Right, so but there is a need for finer burn severity because they may treat them differently. It also depends on the depth and the thickness of the burn as well, but there's a need for finer burn severity here so that we can see how the like lesser extent of the burn severity, if they can develop to be the more severe. And also there's a lack of early predictive marker that can make the clinician to make the decision as well. So our approach is we want to look at early innate immune response as a severity marker. And this is the wound healing. So it's a wound healing process. So here from the injuries, and here's where it heals. And we focus on the inflammation phase. And the reason is that is where most of the innate immune cells like macrophage and neutrophil are recruited. Right? They release the cytokine or chemokines, and they also remove the debris or detect the pathogens. And we think because they are the immune cell type that first see the invader, they may give us the hint to the early prediction. So we work with Anna Hachenlocker and Angela Gibson on this project. We use zebrafish fins burn models, and probably I don't have to convince the audience here the advantage of the zebrafish models. So they have a very similar immunity to humans. That's I'm a little bit surprised by that, but there's evidence that they're very similar in the great extent than I would appreciate. They provide a genetic manipulation tool, which like could help us to study mechanistic after we identify in the marker or things like that. And also one of the things very attractive to me in terms of cell interaction is we can easily than other model to do live imaging because they're transparent. Especially we can check the innate immune cell behavior and can target the population of interest for other profiling using photo convergence. So we work with Anna to generate single cell RNA-seq for zebrafish burn models. So here's the control, like we have the no wounds 3 days after fertilization. You can see here 4 days and 5 days. And as a wound, like we have like after 6 hours post-burn and 24-hour post-burn and 48 hours post-burn. For easily, I'm gonna go time point 1, 2, 3 here. And we did single cell RNA-seq. We identify a neutrophil and macrophage subset, and we identify the burn and the time-specific signature, and we analyze the cell-cell interactions. And single cell for human burn is not existed yet. We are going to generate that, but for now there's no available yet. We're looking at publicly available. And to our surprise, that even the normal RNA-seq is not out there yet. The best data here is actually the microarray data. I call it book of transcriptome, but it's actually the microarray data where they have nearly 800 whole blood sample. But the nice thing is they have a diversity of total body surface areas, so that we can divide the sample into what we call it is moderate and intermediate and high severity and very high severity here. So briefly, we are able to identify 4 neutrophil subsets and 6 macrophage subsets. They seem to have the distinct transcription. So here's the example. Here is this plot called dot plot. And the darker color is the more spread it is, and the size of the cycle is the percentage of the cell that express in a certain subset. We're able to identify 4 neutrophil subset and 6 macrophage subset that have the distinct gene signatures. And they also have some gene ontology pathway enrichment when we use gene set enrichment to compare across the subset. And so there are a number of results come out of that analysis, but I want to present this. When we did the cell-cell communication we asked like between neutrophil and macrophage, how the interactions are changing. One of the things that we found interesting is in terms of the number of statistically significant interactions that we infer. It seems to be the first time point, the second time point are not much different between burn and no wound, and it's that different here. However, the strength of the difference between the cell-cell interaction are very different within the second time point. And the strength here was used the co-expression of ligand in macrophage and receptor in neutrophil. So that's suggesting us a mode change happened in the after 24 hours. And one of the interactions that we identify is the IO6 ligand that expressed in macrophage and the IO6 receptor that expressed in neutrophil. IO6 are expressed in all neutrophil subsets, and IO6 ligand expressed in 2 macrophage subsets. However, it lost in one of the subsets in the burn conditions. And interestingly, this work from Anna when she used the microscope imaging for the IO6 white high and mutant, and you can see like in the mutant neutrophil get lost, it's not recruited. So so this not surprise to her when I saw that, but the extent of IO6 and IO6 in the subset was what we're studying now. So the next question we ask is, can we infer from zebrafish to humans? And what we did is we took the zebrafish genes. We only consider the ones that have human orthologs, and we use the conservation score is pretty conservative here. And we're only looking at the genes that express in human macrophage or on neutrophils, right, using the single cell RNA-seq. And then we correlate them with the severity, which we have around 100, more than 100 for each of the category here. And one of the signature we found here is we found a signature from one neutrophil subset that have positive correlated with the burn severity. And as well, we found one of the macrophage subsets that have negatively correlated with the burn severity. And the co-expression of IO6 and IO6 are also positively correlated. That is in all of the sample. When we're looking at only the blood samples that collected as a day 1, because we want to predict the early immune response, the chance still hold, right? But also we can see the statistical power is a little bit less, but the chance still hold. And if we do randomly, like if we ask whether we observe this by chance, and if we suffer the suffering the TBSA level 4 levels by 100 times, so we don't see that. And I think like we even do 1,000 times, we don't see that at all. So that indicating the signature we found here are potentially significant. And one of the, this is still the signature, right? Each of the signature here is about 10 genes or so. So we still want to see if we can identify the specific gene. And one of the genes we found is ZPR84. And you can see here, ZPR84 are much higher in the burn patients which have the high or very high total body surface areas level. And it's low here. And ZPR84 is one well studied in the neutrophil, so we trying to follow up now. And we also identify it is lesser known gene called ZYZ1. We have the similar pattern. So with that, I'm going to end the first part of my talk where we use zebrafish single cell analysis, identify the cell-cell interactions that have potentially as a marker for human burn severity. We identify the source of the IO6 interaction, and we identify several markers that we're trying to validate and study them mechanistically now. And any question for the first part, or I can move on to the next part? Yes.
So when they took the blood from the human burn patients, from where did they take it from? Do you know?
The blood, like, is just like
You
normal
think you
blood.
were able to get blood, you were able to work with samples?
So that is from public data. The blood sample wasn't from us. It's just the normal blood draw.
Okay.
Yeah, it's not tissue. It's not from the burn. Nothing like that.
Okay, okay, so those people, they did experience burns though, and then the blood was just taken from them.
Yep.
Okay, I see.
Yeah, and that is one of the of the field. And I'm going to tell you some of the future work we do. We're trying to get the sample from burn as well and link with the blood, because that is what missing in the field. Any other question? Yep.
I might well ask this of the second half of your talk as well, but I'll just throw it out there now. What about paralogs? Right, you know, step one is you reduce it down to those genes that are orthologous between zebrafish and penguin. But what can be done about the paralogs? What might we be missing?
That's a great question. We haven't looked at that. Yeah. So we just want to find the one that most similar and most potential reproducible, but that's a good question. We will look at that. Yeah. I even don't know how many parallel between zebrafish and humans, but that's a great question.
If there's a genome duplication in zebrafish, you
Yeah.
gotta think there's a significant
Yeah.
number of them.
And when we do cross species analysis, we found a number of genes in zebrafish, not in fish. So there's one gene called PADI1. That is one of the projects that Anna postdoc is following. We found subsets that express that, and we couldn't find that in human. There's clearly a gap there. Interesting. Okay, I'm gonna start the second part. So I'm gonna talk about immunotherapy in head and neck cancers. So and just briefly, what I'm going to talk about, so there only 20% of patients respond to therapy. And head and neck is one of the typical cancer types that have that response rate. So like if you study melanoma, you have way more sample to do because it nearly 50% of the patient respond. But if you study prostate cancer, for instance, you're going to be very challenged because I don't think immunotherapy work at all for prostate yet. So this is the data that we have, and we use approach we call it system immunology to do the analysis where we have single cell analysis from the treatment naive here. It's not from us, it's from publicly available data. And there's a recent responder and non-responder single cell analysis from Dana-Farber. And there's a book analysis from a clinical trial in Merck and at Yale where they have 120 samples. So these are data from us. We generate single cell analysis in the mouse model of the mouse model that respond and the mouse model that do not respond. And we generate spatial data using 10x Visium and the COSMX data. So one of the findings we found, and I'm going to go into how we find that, is we found the chemokine CXCL9 in two myeloid cell type, dendritic cell and macrophage, and their co-expression and co-localized with the CXCR3 in T cell, which is associated with the immunotherapy response. So how we do it? We reanalyze this data, and seen in this data, they don't have the extent of myeloid cell as we want to see. So we reanalyze it, we identify a greater extent of heterogeneity than before. And we use label transfer methods to predict that in the immunotherapy. So this is pre-treatment and this is post-treatment. This is the work from my grad student, Athena. And this is from the ones that have low response and the ones that have medium or high response. We combine the medium and the high because of the statistic. And we're able to recover the extent of heterogeneity of myeloid cell in the treatment naive in the one with the immunotherapy treatment here. And these are two different data sets. So, and then we perform cell-cell interaction. We ask to what extent the cell-cell interaction happen between myeloid cell and T cell, because here we use T cell as a readout for immunotherapy response, right? Higher CD8, CD4 T cell usually have better chance and that is maybe underestimated because there's subset of T cell as well. But for this purpose, like we want to focus on myeloid cell with a group T cell into CD4, CD8, and the CD4 Treg. And one of the interactions that we found is one of macrophage and dendritic cell subsets that get increased after treatment and much higher in the responder compared to the non-responder. And we're looking into that subset, and we found the CXCL9 that expressed in that macrophage subset and in another dendritic cell subset, while the receptor CXCR3 expressed in T cell. And you can see this is a non-responder. They have interaction, but not as much as in the responder. So each of the line here and their thickness represent how high they are interacting, measured by the co-expression of ligand in one cell type and the receptor in another cell type. So this is the human data that we realize from this publication. And then we are generating our own data where we are able to identify similar subset in mouse model compared to humans. We also found CXCL9 and CXCR3 are only in the mouse that respond to immunotherapy, the K17 knockout here. And the one that is cold tumor have some specific, but CXCL9 are in the ones that respond to immunotherapy. And similarly, CXCL9 is spread in myeloid cell and CXCR3 is spread in T cell and NK cell. So that makes it wonder, like maybe CXCL9, CXCR3 can predict the immune checkpoint response. We're looking at clinical trials that published in Science in 2018. They only have 18 gene panel because those samples are from like the frozen sample that they bind for a long time, I think. And they published in 2018, but the data for you earlier. And they have a lot of samples. We are able to compare CXCL9 are much higher in the responder compared to non-responder. And we have about total 100 samples here. Whereas the CD274, the PD-L1 that were used in clinic is not we still see the change here. Usually the PD-L1 high patient is the one who more likely to respond. And PD-L1 is one of the criteria that help clinicians to make the suggestion to the patient whether they should go to immunotherapy. But we don't see much different here. We're looking at the data set from Yale. They have less sample size, but they have way more genes. They have 18,000 gene panel. It's still the nanostring technology. And you can see like CXCL9, we don't see the difference anymore, but you can see it's very, very, it's very, very variable between the responder and also the non-responder also we have more responder than the responder. But we see CXCL3 is a little bit higher in the responder, right? There's no CXCL3 in this 18 gene panel, so we cannot do that comparison. And in literature, recently, people study CXCL9 macrophage, CXCR3 T cell, and arguing they are crucial for the immunotherapy response, but they never link them together. Usually they study them separately. There's a number of papers in different mouse model here. So that makes it wonder, like whether we can use spatial data to refine this analysis and asking if CXCL9 and CXCR3 are located in the same locations in tissues. So we use a technology called 10x Visium, and for some of you who may not familiar, that can provide up to 18,000 genes. And now I think they have the updated versions called Sciasis. That probably can provide way more. It can work very well with the embedded tissues like FFPE or OCT embedded. And each of this, they can profile up to 5,000 spots within the tissue. Each of the spot can have like 10 cells, 20 cells, some of the spot they have 30 cells. They don't have the single cell resolution. We use the single cell type annotation that we identify from single cell here. And again, we use the label transverse deconvolution and ask to what extent we see epithelial tumor cell, stromal fibroblast, and immune CD45 cell. And you can see like it the deconvolution. Well, I don't have it here, but we consult with the pathologist when they're looking at the HNE, and it largely agree. But like we didn't go to specific cell type yet, right? We just go to for the 3 major cell type, so that's not very challenging. So, and we're looking at CXCL9 and CXCL3 in the immune cell. And here an example of 2 responder and 2 non-responder. You can see like this T cell have a lot of immune cell based on our estimations, and they have a little bit less. So, but both of the patients are response. And they have more immune cell, and they also have less, and those 2 patients do not respond. And when we use the co-expression score, and we see like there's more colloqui of CXCL9, CXCL3 in the immune cells from the patient who responds and the non-respond. So you can see like this is not a lot, right? But there seem to be like the CXCL9 immune cell and the CXCL3, and we know that where they're from single cell are colloqui in the T cell. So quantitatively, we asked, because we have a little bit more than 4 samples, so like we have about 11 samples, but we have to remove 3 because of the low immune and marker cells that we can recover. We asked what are the difference between the frequency of the immune positive spot defined by the amount of immune cell that have expressed both CXCL9 and CXCL3 out of all the immune cells. And what we see is in the 4 responders, they are around 30%, but one of the samples have surprisingly like more than 80%, and most of the non-responder have the low. Also, like we need to increase the sample and do more independent validation to validate this, but the result is encouraging. So currently we are trying to do more analysis to identify more interactions, and also we want to identify the interacting neighborhood and study the mechanistic of those interactions that we found by either in vitro or in vivo model, where we can blocking the ligand or knock out the receptor. So that will be the second. Like, any question before I go to the last part? Any question? Okay, so I'm going to talk about the last part, and you can ask a question after that as well. So now I'm going to show you how we use the same approach to identify the early marker in ovarian cancer detection. So as all you know, sadly, most of the ovarian cancer patients was found very late. That's why the survival rate for this disease is very devastating. Over the last 10 years, I don't think there's a significant improvement yet. So here we want to look at tissue origin, which is the fallopian tube. So actually, I'm going to show you this. So ovarian cancer do not origin from ovary epithelial itself, but from the fallopian tubes. There's a histology, every data showing that where they were able to detect the P53. So as you know, that is one of the cancer hallmark in the fallopian tube of the cancer patient, but not in the ovary. And there are a number of genomic and transcriptome sequencing data support this hypothesis, including our data where we analyze using machine learning to predict the signature of the fallopian tube and ovary, the FT6 and OV6 here with the TCGA data. So we generate single cell RNA-seq and book RNA-seq for ovary epithelia and fallopian tube epithelia and use that signature to predict in TCGA data. And you can see like fallopian tube have more signature in the chaining set as well as the validation set. And also we publish a single cell data to support that as well. And now I'm going to tell you how we characterize the single cell of the human fallopian tube. So we published papers in 2021 to characterize the epithelia, which is two subset here. Now I'm going to tell you the work from my students who characterize the rest, which is the fibroblast and stromal cell here, T cell and NK cell, and the myeloid cells. So briefly, what he found is he found the ratio of two immune cells, so-called macrophage and monocyte. They are chained between the benign tissue. This is a benign fallopian tube. So that is from the patient who do not have cancers. And you can see like we see way more macrophage than monocyte. And this is the fallopian tube from the patient who have germline BRCA1 and BRCA2. They are known to have the high risk to develop cancer. And we start seeing 4 of them look more like cancer, and 6 or 7 of them look more like the normal phenotype. And the adjacent normal is a little bit higher than the fallopian tube here, but it's still in the low range, meaning they have a little bit more monocytes than macrophage. And then it increased, especially with the high-grade cell, you can see like way more macrophage than monocyte. This is an example of the data set that have adjacent normal in tumor, three patients. And you can see this amount of macrophage in normal, and this amount of macrophage in tumor. So the monocyte seem to be in both normal and tumor, but the ratio are clearly different between normal and tumor here. And one of the things that Josh did is he identifies the interaction of monocyte and macrophage with other cell types. And what he found is they largely overlapping. 83% of them are similar, and that's not surprising because they are both monocytic genes. Recently with a single cell, we more appreciate that. But if you talk with some of the immunologists, they still believe like most of the monocytic genes in the tissue are macrophage. There's no monocyte in macrophage. Monocyte usually linked with the blood sample in the circulation. In circulation, it doesn't have macrophage. That is a tissue resident cell, immune cell type in tissue, right? But now we have evidence they are in both. And there are about 16 monocyte specific interactions and 22 macrophage specific. And if we use those interactions and compute the score in the match normal and tumor, which we take from this data, so they are normal in tumor ovary. And you can see in tumor, the macrophage specific are more enriched in tumor, and the monocyte specific interaction are more in the benign ovarian tissues. But the ones that are in both some of them are more specific, but both of them are associated with both. One issue is if you notice here, the interaction that's specific for either macrophage monocyte is very low compared to the one just said, right? So that is one of the challenge. Like if we want to take this and then develop a biomarker, that's going to be a very challenge because the expression, at least in the RNA, what we detect here is very low, right? So that is the current one we have, and we're trying to work on that. Another piece of data, not so the data, but we found that the link of monocyte and macrophage from benign to the patient who have the high risk that carry BRCA1, BRCA2, and the cancers are linked with the dysfunctional T and NK cell. So more monocyte, less dysfunctional and T and NK cell, more macrophage, more dysfunctional and T and NK cell. So we think the tissue microenvironment already seeding a pro-tumoral in the tissue before even like before they develop to be the cancers where the cancer develop in ovarian. So we use multiplex immunofluorescence to validate the presence of monocyte in benign fallopian tube because most of the paper public out there, they don't report the presence of monocyte. And we found that in single cell, but we want to see that in as a protein level as well. And this is an example where you see like this is a monocyte marker, and we saw many reasons that have that express that as a protein level, and this is an example in tumor. So with that, I'm gonna end the third part. So hopefully I was able to show you some of the example. I need to summarize it first. So we take the data. So one of the good thing with doing the hybrid research is we start this project during the pandemic. None of the data is out. All of the data here is publicly available data, right? So like if we have to generate the data ourselves, probably this project not gonna happen. We are able to identify the marker based on the monocyte macrophage that potentially in associated with the early development of cancer in ovarian cancer. Currently, we are trying to validate them for more sample and using protein imaging technique. And also we want to do spatial omics for the sample that carry BRCA1 and 2, because we are very curious about what makes those patients here different from patients here. Those patients have their fallopian tube removed. We never know whether they're going to develop cancer or not. I mean, we're not allowed to know as well. So they may never develop cancer because they take some like high risk surgery where they don't want to have that chance. So with that, hopefully I was able to show you the 3 examples where we use single cell and spatial data to identify the potential marker in human burn severity in response to immunotherapy and in early cancer development. And the ongoing and future work is we want to incorporate across species, especially for the burn project. We want to ask, like, if we are able to identify the single cell and spatial atlas of the cell interaction across the time points using the human, where we collect both blood and tissues. And then we're gonna use the mouse model from collaborator and zebrafish from collaborator to see what translates and what's not. And we also want to study mechanistically of how it happens. And with that, I will end my talks with the acknowledgement. So I saw the work from ovarian cancer from Zoss, a student in CMP program, the head and neck cancer project from Athena, also from CMP, and the burn project from Pat, from BDS in collaboration with Iran, a postdoc from Anna Lab. And we just got a grant to study burn and time cost and all of the data. We have a lot of data in genomic, spatial data and imaging, and recruiting if you're interested, someone, some student or postdoc interested in putting the data together. So shoot me an email. And this is our funding. Thank you for the attention, and I'm happy to take any question.
I have a question that maybe is a little less on the immunology side and more on the genetic side. So it seems like you were starting to identify the markers of what tumors will respond to immunotherapy for.
Yeah.
But I'm wondering what the basis of the response is and how, so I know there are differences in cancer, tumor type.
Yeah.
Some tumor types respond and others don't. Do you think there's a genetic basis? And actually, maybe this was the BRCA1 stuff at the end. Is there a genetic basis in the, I mean, I guess either in the tumor, but maybe in the post, you know, patient,
Yeah.
germline of the patient.
Yeah.
That determines if they'll respond.
So usually the tumor with a high mutation burden have more chance to respond to immunotherapy. And we just see a talk that actually from a visitors today who's showing the BRCA1 patient is more respond. They don't know why yet, but I don't.
So were you showing at the end, the BRCA,
Yeah.
the benign tumor
Yeah.
or something?
But that is not immunotherapy. That is a cancer development actually.
That's a native immune response.
Yeah.
Yeah.
So this is a patient who don't have cancer. This is also from the patient who do not have cancer. They don't have a germline, and then they remove their fallopian tube. That's why we get the tissue, right? They are not cancer patients, but we found some of them have look more like tumor, right? But of course we cannot predict it. We don't want anyone get cancer, but that is our hypothesis. If the patient carries this germline mutation and have the immune phenotype like we saw, so that is a pro-tumoral immune phenotype in our opinion, but we don't have the evidence. Don't take my word for that. That's our hypothesis. Yeah.
So I think I missed something pretty fundamental in your talk. I still don't understand how you can determine cell-cell interactions. Are you sequencing groups of cells and looking for cells that are commonly found in groups?
We sequence everything and we identify the cell type. And so we identify the different cell type here.
Right.
And we ask if ligand is spread in one cell type and their corresponding receptor is spread in other cell type. And if they are co-expressed, so they likely interacting.
Oh, so you're not actually determining a physical interaction?
No, not yet. The imaging data maybe, yeah.
All based on literature, and you're right, there are known ligands.
There's a known ligand.
receptors, And you're asking whether what are the cells that are expressing the ligand, what are the cells expressing the receptor, and you're assuming that they are touching.
Yeah, yeah, and then we have to use imaging data to show that. But that's really a very good question, because we have a rotation student where I asked her like, is there any like tabs in that look like the known ligand receptor? If so, are they maybe de novo interaction? Yeah, we're trying to work on that as well, but currently I don't see any tool right now that do de novo ligand receptor cell interaction yet.
Well, I'm just thinking if you could somehow sort cells in such a way that you have small groups, one or two to let's say 10 cells, and sequence those groups.
Yeah.
Then you know those cells are
already interact. Yeah, there's a technology developed by UW-Madison Lab from Weizmann Institute. They call it PIC6. And BD developed a technology where they call it image-based sorter. They really see the cell attack each other in image, and then they take that for sequencing. We don't do that here yet. Yeah, but that would be the next, and that would be truly interactive.
A follow-up on that, does that give you more information than the spatial transcriptomics? I know Visium doesn't have very good resolution. But the PIC6 that you just mentioned.
Yeah.
Does that give you a different level of information of the type of contact than you did from spatial?
Well, they public an application in the interaction between dendritic cell and T cell, and they discover some like unknown interaction, especially in the immunotherapy context. But they also discover most of them are also known that who already found described literature by other technologies. But that is the purpose, right? They want to discover the unknown one because they already know the cell physically interacted. We don't have that here yet.
But they may not be with a ligand receptor connection. Is that what the distinction is?
So you mean like the interactions that do not use ligand and receptor?
Can you get that information just out of the spatial transcriptomics?
Yeah, that's what we are also trying to do now. But we have to assume the cells that close each other are more interacting, And one of the things that, so we have a project where we plotting the distance in the spatial data with the expression. We see some of the interaction if we use the conventional score that we developed here. They also have the similar when they are not close to each other. So I think there's a short range and long range interaction, and they may work as a network rather than just individual. Yeah. But that is there. And but the answer is yes, that should be from spatial data. Yeah. Single cell, we're not gonna have that power. Yeah. Because for single cell, like to re-emphasize what you misunderstood is we just ask if they co-express and we hypothesize they interacting. Yep.
So I wasn't sure, you said you haven't had a lot of time to gather data, but do you have any comparable images to show us?
Yeah, the IO6 are actually the confocal ones. Yeah, but that is not our data. It's from Anna Hattenlocker lab for zebrafish. Yeah, this one actually confocal. Yeah, but it's not from us. Yeah, so it's just like encouraging that's what we found the single cell also found in the confocal. But IO6 are, like you said, like it's a known one. Everyone studies that already. Hopefully we are able to validate some of this. Yeah, that's only, but we don't have the evidence for the novel interaction that we found here and whether or not we can validate them by the microscopy. That is one of the goals.
Thank you.
Okay, if there are no further questions, let's thank the.
Thank you.